How's it going, everyone? My name's David. Um, I decided to take a look at the intensity of schistosoma mansoni infection and fecal calprotectin in preschool-age children in the Lake Albert region of Uganda. So a little bit of background we'll start off with on schistosomiasis. It's a waterborne parasitic infection that results in both acute and chronic infection. Um, Acute infection can be as simple as a itchy red rash, where the parasite enters the skin. As far as chronic infection, it can infect a myriad of different organs and organ systems, including the brain, the lungs, spleen. It can result in poor neurodevelopment and stunted growth in children, and impacts over 200 million people worldwide, and is the second most economically impactful parasitic infection in the entire world, second only to malaria. And Schistosoma mansoni is the species of blood fluke responsible for infections in Uganda. It has a national prevalence of about 25.6%, and in children aged 2 to 4, it has a prevalence of about 36.1%. So an extremely high burden, particularly in the Lake Albert region. So now we'll briefly discuss the life cycle of Schistosoma mansoni, starting with its presence in freshwater snails. So it infects these snails, and these snails release sporocytes into the water that become these Croceriae, which are essentially like small worms that penetrate the skin of human hosts and infect them. The human hosts may be using the water, for example, with Lake Albert. It's used for recreation. It's used to gather water for homes. It's used for bathing. It's used for defecation, urination. It's used for work, like fishermen fishing. So there's a lot of exposure to these little Croceriae, which then enter the skin. They develop into full-grown worms that have this lovely romantic relationship. They produce eggs in the intestine, which burrow into the intestine. And once implanted there, they cause inflammation of the intestine, which brings us into our next topic, which leads us to our next segment of my project, which is environmental enteric dysfunction. So environmental enteric dysfunction is a chronic disorder of the small intestine, which there's not a ton known about it. It's only really become relevant in the last 10 to 20 years. It's distinguished by abnormal changes in the function and structure of the gut, and it results in reduction of absorptive surface area and function of the intestinal barrier. And it is presumed that this is related to increased inflammation of the intestine, which is starting to make sense with my prior discussion on schistosomiasis and what it does inside the human body. It diminishes the ability of the small intestine to absorb nutrients, and has a strong relationship with stunting. It's associated with poor water and sanitation, and found extensively in low and middle income countries. Calprotectin is one of a few biomarkers that's utilized for identifying EDD, or as a potential identifying marker for EED. It is a binding protein primarily found within neutrophils, and it's found within feces due to the inflammatory process. So my question was, do preschool-age children with greater burdens of schistosoma mansoni infection, measured in eggs per gram, have higher levels of fecal calprotectin, which would be indicative of EED? So this was a cross-sectional study that used data collection from an ongoing NIH-funded phase 2 randomized clinical trial examining protoquantal dosing. There were 352 preschool-age children enrolled that lived in the Lake Albert region. These were enrolled by local healthcare workers and research assistants working on the project. Ages ranged from 12 to 48 months. They all had active schistosoma mansoni infection, but were otherwise considered healthy. And I was able to utilize the data of 296 of these individuals. Some were left out due to missing socioeconomic information or test results that I wanted to utilize. And speaking of test results, blood and fecal testing was utilized to obtain results for malaria, HIV, intensity of infection, the EPG value I spoke about, and fecal calprotectin. And this was all performed at the Uganda Research Unit in Entebbe, Uganda. All socioeconomic information was collected via survey of patient parents by research assistants during clinical visits at local health facilities. So I started off with a DAG to kind of visualize the relationship between all these factors. So I looked at parental education, mother and father, toilet location, age, malaria co-infection, home water source, and HIV co-infection, and how they impacted EPG values and fecal calprotectin values, and also intestinal inflammation. So as far as some additional methods, I utilized SAS 9.4 as the software program. I used simple linear regression to test if increased EPG significantly predicted increased fecal calprotectin levels. A logarithmic transformation was then utilized on the dependent variable as it was not normally distributed. And once normality was established and a potentially significant relationship was identified, a multiple linear regression model was run, which included additional biologic and socioeconomic variables of interest. So these were just, this is my table 1, and we can look at some of the demographics and characteristics of the population that I was looking at. As you can see, so one thing I want to point out here is that the source of drinking water was the only multiple choice answer. So as you can see, 289 individuals utilized the lake out of the 296, and then others also would utilize boreholes, piped water, and looks like one individual utilized a hand pump. So the results, ultimately I found out that it was increased X per gram did not significantly predict increased fecal calprotectin in preschool age children with schistosomiasis. It was found that age and toilet type were significant predictors of fecal calprotectin in preschool age children, which, as we'll see in the discussion, is supported by literature. Fecal calprotectin does tend to increase with age and then tapers off in the teen years into adulthood. And then toilet tape was something that I did kind of, it was challenging finding literature as to why that was significant. My thought was that it could be related to increased exposure to pathogens at shared toilet sites, which a number of individuals who responded to those surveys noted that they utilized shared toilets. Although my methods did not yield a significant relationship, other methods could be utilized to further investigate this question, such as using a spline or taking account for a threshold effect. Additionally, some other biomarkers, comorbidities, and socioeconomic factors could be analyzed and gathered, and I was kind of like limited by the survey, and may also benefit from the inclusion of preschool age children without schistosoma mansoni. So yeah, overall, interesting project to work on. Didn't get the result that I wanted, but still a lot of fun. So if you guys have any questions, I would encourage you to post in the discussion. Looking forward to answering them. And I just wanted to acknowledge Dr. Jennifer Friedman and Susanna Colt, who were both incredibly helpful throughout this process, providing me with the data and all of the information I could ever need on EED and schistosomiasis. So thank you all. Have a good one.